Hi, I'm Dr. Shankara Cherry. I'm a general practitioner from South Africa and a frontline doctor, and a panelist on the Hidden Drivers of Post-COVID Dysfunction conference. I look forward to seeing you all at the conference.
Hello, my name is Brigitte König. I'm from Germany, and I'm a professor of microbiology and infectious diseases. And I look forward to see you at the conference.
Hello, my name is Marcus Klopp. I'm the founder and director of the Afrezia Center, the world's first purpose-built long COVID and post-vac and chronic illness clinic in Cyprus. And I'm looking very much forward to see you at the conference.
Hello everyone, I'm Risha Pretorius. I'm from Stellenbosch University. I'm a professor in the Department of Physiological Sciences, and I will be sharing some of our insights into microclotting and vascular health in long COVID, and I'm looking very much forward to seeing you at the conference.
Over the past few years, I have been focused on a topic that seems to largely have been ignored by the scientific community, and it's that of embalmer's clot. Because my point is that even though I don't understand what the exact mechanism is behind it, it's very unusual. We have not seen this before. Part of that work from a scientific point of view is to therefore try and make sense of the science. Not many scientists have been willing to look at all at anything to do with clots. However, there is one who is not only willing to do the research, but willing to talk about the research and focus at it from the scientific perspective. I'll be asking him some of the hard questions that you may be curious about, and we will hear him explain it from a scientific point of view. So without further ado, let's bring in Professor Douglas Kell. Thank you very much for taking on this topic, Doug. How are you?
Well, nice to chat again. We did one of these before, and I was extremely impressed how well informed you were about all of these matters and asked jolly good questions.
Excellent. I'll ask you, for the purpose of the audience, if you could just introduce yourself and your background.
So I'd probably normally say I'm a systems biologist. I was a biochemist originally, and for these purposes, we've been looking at long COVID, along with my long-standing collaborator, Professor Risha Pretorius from Stellenbosch University in South Africa.
Excellent. And so what is it that has been your fascination with regards to clots? Because you clearly do a lot of research on it. Where did that interest come from? Is it just something you came up on in terms of your science and so on?
So the origin is actually, I used to work on, and still do a bit, work on the metabolism of iron, the metal iron. And I wrote a big review, a very big review. It was a whole year's worth of weekends of 2,500 references, called Iron Behaving Badly, showing that it was involved in all kinds of diseases. And I read a paper by Risha Pretorius from Pretoria, who'd been looking at the effect of iron on blood clotting, and it caused it to clot into this highly anomalous form, which he observed under the electron microscope. So normally, if you look at a blood clot in an electron microscope, it looks like spaghetti, like in spaghetti bolognese. Whereas if you looked at the clot formed after you've added iron and initiated the clotting by adding thrombin to blood or fibrinogen, which it contains, you got something that at the time they referred to as dense matted deposits, which was just an unholy mess that looked like if you parboiled spaghetti and let it clag together into a rather sticky, yucky mess, that's what it looked like. And so since I was studying iron too, we started talking together on the Skype and decided to work together. And it's been a highly productive collaboration with over 60 papers now, where we've looked at all kinds of aspects of clotting. Fast forwarding away from the electron microscope, when we discovered that the clots were amyloid in nature, we'll come to what that means in a bit, and could therefore be stained with a fluorogenic stain. So you could see them much more easily in an optical microscope with fluorescence, which is much more high throughput than looking in an electron microscope. So we studied this in a lot of diseases, and of course COVID comes along, and it turns out to be highly relevant to both acute and long COVID and other post-viral diseases such as ME/CFS and so forth. But most recently, what catalyzed this interview with Philip is our discovery that the clots removed from people who've had an ischemic stroke are also amyloid in character. And one of the properties, of course, is it makes them much more resistant to being got rid of by the normal methods of fibrinolysis, as we say, getting rid of the clots. And normally, blood is clotting and unclotting the whole time, so that if you cut your finger or whatever, the clot will happen immediately. You don't have to wait for genes to be turned on and stuff like that. And then once it's done its job, it's called plasmin, an enzyme that removes the clot, will normally remove the clot. But our amyloid type clots, we call them fibrinolytic microclots, are much more resistant than the normal clots to being got rid of. And so they slosh around.
I just wanted to pause you here a second. And I just wanted to clarify, because people will be curious about this. Are the clots that you are seeing now, even with regards to the microclotting with long COVID compared to ME/CFS prior to the pandemic, are they about the same? Or are they different in terms of the number or the characteristic, or are they about the same as it was?
Oh, so it's certainly gone up as a result of the pandemic, if that was the question.
Yes.
And because when we started, the normal controls had essentially zero clots, very close to these microclots. Whereas after the pandemic, and also when people got vaccinated, their numbers would go up in the controls quite significantly. And then if you got better or you didn't get long COVID, then for the vaccinated people, they went down. That was published by Caroline Dalton. Whereas for the people with long COVID, they stayed up. And the point of the microclots is they go round the bloodstream, blocking up capillaries, stopping red blood cells bringing oxygen to tissues, and thereby giving a pretty straightforward explanation for the hundreds of symptoms of long COVID. So you might think a unitary explanation would be very difficult to imagine given how many symptoms there are. But as it turns out, they can explain all kinds of things such as fatigue and brain fog, post-exertional malaise, which is slightly different. We could go into that, and postural orthostatic tachycardia syndrome, or POTS, and even atrial fibrillation. All kinds of symptoms can be explained by these particulate microclots being stuck in your system, not being got rid of, which may seem surprising, but if you do get rid of them, people get better. So it's pretty clear that this is part of the story.
I think it's important, that we do a little bit of a focus on what it is that you had done recently in your paper, and then we'll come back to some of these principles, because I wanted to talk to you because this paper, which is in preprint and you said is due to be published soon, stood out to me as a very important piece of work. That the clots removed from ischemic stroke patients by mechanical thrombectomy are amyloid in nature. This is a relatively recent study, and I'm assuming, therefore, that these clots have occurred in the past few years.
Some of them are pre and some of them are post-pandemic, I think. But and of course, what we hope is that people who've stored clots elsewhere, and we've been talking to some from long before the pandemic, will study them in the same way as us. So what we did in this paper, so I'll just explain thrombectomy for people who mechanical thrombectomy. If you get an ischemic stroke, which is where a clot blocks up an important artery in the brain, you give an enzyme, or you try to give an enzyme that is supposed to get rid of the clots, which is an activator of plasminogen, called recombinant plasminogen activator, tissue plasminogen activator, and you there's a picture thrombolytic drug, which is supposed to chop the thing up. It's not terribly effective, if truth be told, but it has some effects. But obviously, if it worked completely, you wouldn't have to get rid of the clot. So what happens in mechanical thrombectomy is, whether you use this thrombolytic drug or not, you pass a small tube up the femoral artery, which is in your thigh, all the way up, following it with imaging while you're doing it. And inside the little pipe is an even smaller net, like in a lacrosse stick, if you will. And then when you get to the clot, the lacrosse stick comes out, pulls the clot backwards out, removes it, and then you can pull it all the way back down the pipe and remove the pipe. So that's mechanical thrombectomy, and it's considered to be the kind of standard of care, especially if you can get to the stroke quickly after it's happened. And so normally people collect these clots and essentially throw them in the dustbin, whereas in this case, our collaborator Alakendu Sekar and his colleagues at the Walton in Liverpool collected them and put them in a like a blood bank, but in this case a clot bank. And the way you do that actually is you embed them in wax, and then they can sit there forever. So we obtained some of these. Sorry.
Yeah, just saying, you said something very important. So you have got clots from pre-pandemic, and you've got clots from post-pandemic. And
so let me stress that the numbers we could do at this time were extremely small. So I'm not keen to overgeneralize at this stage. What we want to do, of course, is to go on to do much larger numbers, studying exactly pre and post pandemic and many other properties besides what we discovered by staining them. So what we did was we stained them with the amyloid stain, and boy, they lit up like Christmas trees. And I'm sure you've got some pictures of that.
I'm going to show you that picture in a minute, but I think that this is a very interesting question when we therefore think about it, because if you have gotten more clots that have been collected pre-pandemic, it would be quite fascinating to see whether or not the morphology in the detail of these clots are exactly the same before and after, because that could be quite valuable information on its own.
So we're keen to look at all of the properties, the morphology, the proteomics, different kinds of stain, susceptibility to proteases. There's a long list of stuff, and Raman properties, and recently, as we'll get on to.
This is here. This is
been looking at this. Yeah.
This is an example of one of the images. Yes, I was just
so we did 8 images in this study. We had 8 patients initially, and if you're amyloid in nature, you're bright green, and if you're not amyloid in this imagery, you're dark. And we'll, I'm sure, look at another one that combines bright field with amyloid too. So when we get these clots, first of all, we have to slice them so they're thin enough to look through in a microscope. And that was done by our colleagues in the Liverpool Shared Research Facility histology lab. And then you remove the wax with various solvents, starting with xylene and ending in alcohol. And then you can stain them because they're watery, so you can treat them like that. So that is what we've done. We've got these thin slices of the clots, and you can see, the diameter of these things is almost a millimeter or more in some cases. So these are essentially slices through the clot that's been removed from these poor patients. And in all cases, there's a lot of amyloid, some more than others. Like F is mostly in the shell, and it was known before that the shell was different, but not why it was different and how it was different. And astonishingly, no one had ever thought to stain for amyloid. We were the first people to do it, and that's what we found.
So when you say you were the first people to stain for amyloid, nobody had ever looked for this in clots before?
Well, we haven't found anyone who published it, and some of my co-authors are very well-respected, long-serving stroke and clot experts, and would have told me, and the referees in the published or to-be-published paper would have definitely said, oh, this stuff's well-known if it had been, but obviously it's easy enough to find out if it's known or not, and it wasn't. Or not in published form.
Not in published form. So we are then seeing these characteristics with regards to amyloid. Are you able to explain in a simple way what is amyloid and why it's relevant to these clots?
So to understand amyloid, you have to understand first protein folding. And there's a classic experiment going back to 1970s by Christian Anfinsen, who took an ordinary protein called ribonuclease, unfolded it by adding some nasty things that are a bit like detergents. And so it, instead of being folded up, became completely unfolded. Then he removed the nasty detergents, which are actually urea and guanidinium, and it folded back up to the same shape that it had when it came off the ribosome, i.e. when it was synthesized originally. And that led to two conclusions, one of which was correct and one of which wasn't. And the first conclusion is that in order to fold up, all of that information is held in the sequence of amino acids in the protein. Nothing else was needed. It would fold up on its own. That was true, and it got him a Nobel Prize. There was then an assumption made that the state into which it folded was the state of lowest free energy. And that always had to be an assumption, because if the protein is 300 amino acids, and each amino acid could take 10 positions, that gives you 10 to the power 300. And the lifetime of the universe in seconds is 10 to the 17. So clearly you couldn't know that it was the state of lowest free energy. It could have just been the most easily accessible state. And that, it turns out, is the case. So most proteins do not fold up into their form of lowest free energy, but they can do if they meet the right catalysts. And one of these forms is generically known as amyloid. It has a lot of so-called cross-beta sheets in it, which cause it to have this higher stability, hence lower free energy. And many such proteins are known associated with diseases such as Alzheimer's and Parkinson's, and there are about 50 amyloidoses. We're not talking about that, but they all have this cross-beta sheet. But even ordinary proteins like insulin and lysozyme, if you treat them slightly nastily by sticking them at pH 3, they will form this amyloid form too. No change in sequence, just a move from a state of higher free energy to a state of lower free energy, getting through, or in this case, underneath the energy barrier that normally stops you doing that. And what we discovered, sorry.
Yes, I wanted to ask you, as you're on that point of amyloid, because tied together with it is, what is the difference between an amyloid and a prion protein? And I've got this image here to try and see if I could have captured at that time, where you get a similar aggregation with prions. Why is it different with amyloid?
So the short answer is it isn't different at all. So if you look on the left-hand side where you've got ribbons there in helices, they're representing alpha helices. And now in the middle, where it's gone to the anomalous or abnormal or rogue form, it's sometimes called, where you've got those ribbons there, they're so-called beta sheets. So there is no change in sequences. It's clear the green bit is the green bit and the red bit is the red bit. But they have changed their conformation into, in this case, anti-parallel beta sheets. Sometimes they're parallel, and they map onto each other in the way described with these so-called cross beta motifs, and they are much more stable than the form on the left-hand side. And importantly, they can also, in the case of the prions in particular, catalyze the transfer of PRPC, in this case, to PRPSC. And it's very active. So a tiny, tiny amount of PRPSC will cause millions of molecules of PRPC to flip over into the abnormal form. And the abnormal form is insoluble. And in this case, it's self-seeding, but you can also have cross-seeding, where you make a fiber composed of all sorts of things that have joined the party and flipped into this amyloid form. And as you can see, they are stacking in that way, as in the picture.
And so when we think about the amyloid clots that you had seen, they also have a similar stacking pattern. It's just that they are not prions by converting other proteins into abnormal shapes.
Oh, they do convert other proteins into abnormal shapes, because we know that in a normal clot, which in an electron microscope looks like the spaghetti, as I said. You can look at the protein content of that, and as well as the fibrin, which is what the clot is mostly made of, it traps other things, and you can measure what the other things are. And the other things have been widely measured by many groups, essentially reflect the concentration of soluble proteins in the plasma from where the clotting took place. Our anomalous fibrinolyd microclots are not like that at all. There are many proteins that are abundant in plasma that are not in the microclots. And there are equally many proteins that are far from abundant in the plasma that are highly enriched in the microclots. And the common denominator of those proteins that are highly enriched is that they're highly prone to flipping into an amyloid form. And you can calculate this. There are many web servers to allow you this by looking at their sequence. So certain kinds of sequences are prone to amyloidogenesis, others are resistant to it. But the amyloid form is always much more stable and indeed is much more resistant to proteases. In the case of prions, the test for the PRPSC is to add a very potent protease called proteinase K. It's even in some shampoos for degrading your hair to get rid of the split ends. So it's a very potent protease. It gets rid of everything except the PRPSC protein, and that is your assay for PRPSC. And you can use these tiny amounts of PRPSC to flip the normal form into the anomalous form, and you can actually use that as an assay for how much PRPSC is there.
So I mean, so this is quite concerning at the same time as fascinating. And I'm going to bring you, because we know that this work that you've been doing with the clots in stroke patients are definitely showing amyloid characteristics. I want to bring you to that piece of work that recently came out, because I'm interested to hear your thoughts and how this correlates with the research that you've done. And this is from Kevin McCairn, where he is talking about the amyloid fibrin aggregates in some of the clots that he has been looking at. So the first point is, we can assume from a scientific point of view that these clots are real.
I have no reason to doubt it.
Yeah. And therefore, when he looks at his characteristics with regards to these clots, and I think that there are some specific characteristics that I was fascinated by. I'll show this to you here again. This is a close-up of it here. This is the amyloid characteristic just under the microscope. This is what it looks like, he says, when they cut through it. So they use a scalpel, so it's solid, it's all together. Then the bit that fascinated me was when they definitely saw lots of amyloid based on the thioflavin, which is very similar, quite diffuse, and critically
with some hot spots too, interestingly.
With some hot spots. And then critically, when we look at the 1000 times, this is the surface of it, and then when you zoom in at 5000, this is what it looks like. What can you say? I mean, what the heck is this?
Well, to answer that, you do proteomics to find out what else is in it besides the assumption is that it's fibrin because it's in blood, and you need fibrin to make clots. I don't think they actually tested that, but I think it's it's got to be that. And fibrin is the precursor of fibrin is the second most abundant protein in plasma at 2 to 4 grams a liter. So what it is clearly some kind of jelly made up of fibrin plus who knows what until they measure it. But the main point is it's insoluble. It's a clot, and yeah. That is the point of what a clot is. It's insoluble. And indeed the prions and so forth. And the way blood clotting happens is that you have a sort of knobs and stalks going along this way. So a blood clot is fibrinogen is about 45 by 5 nanometers, and then it bolts itself together. And on that scale, if my index finger is one molecule of fibrinogen, it's sort of this size, typically, a blood clot of the normal kind, the spaghetti, and it's going that way. Whereas in the beta sheet version, the beta sheets are going this way to make the fibrin. So it's a very different phenomenon. And then molecules, individual fibers wrap around each other like you would make a rope.
And why do you think that there has been so little interest in clots, even before the pandemic? Because as you said, some of the work that you're doing had not been done previously. Is it that we have just made assumptions about what clots are, how they form, what their characteristics are, and we have never needed to look at it in any more detail?
I could quote Andre Geim, who got a Nobel Prize for graphene, to the effect that everyone could read the literature, but not everyone does. So it's hard to know. And you often get asked, anyone who makes a scientific discovery says, why did no one else think of that? And I sometimes give the Damien Hirst answer, when Damien Hirst, the artist, was told, well, anyone could have put a shark in a tank of formalin. His answer was, yes, but they didn't, did they? So we did discover this stuff. Why other people didn't, or maybe they did, who knows? But they didn't publish it.
So I'm going to show you a few images here. And this is just, in a sense, following on from what you said in terms of what is in some of these clots. And this was some work that was done previously. And they had 4 clots, and they did the proteomics on it. So this was a teenager. And the primary thing was hemoglobin subunit beta.
Okay.
And so it seems as though it was they were not in equivalent concentrations, alpha and beta as well. Fibrinogen, as you can see, purple, yep, was still high. Myeloperoxidase. So some of that makes sense with the proteins, but between
I should say then, myeloperoxidase is a constituent of what are called NETs, which is neutrophil extracellular traps, and we know that our microclots also contain NETs. There's a preprint out on that, although we've had some trouble with the journal getting it out properly.
And just a quick question with the myeloperoxidase, is it possible that it could also be macrophage extracellular traps? Would we be able to tell the difference between
I don't know. I don't know, because I don't know scientifically. This is not my area.
Okay.
The nets we did with someone else who's a net expert. But I'm just looking at these things to see if there's anything. Fibronectin makes some sort of sense. Anyway, the point is, studying the proteome of these things gives you lots and lots of information about how they were made, where they went.
This was another page. So they're not exactly the same. This is what's fascinating.
Oh no, they're never going to be exactly the same. That's because we're pretty sure that the precise nature of the clot. I mean, and indeed, in the images we saw from our work, every clot looked different percentage amyloid, different morphologies. So we think that when the clotting happens, it essentially integrates all of the small molecules, and indeed proteins, that are present at the time of the clotting. Although in this case, it's entirely possible, in fact, we even think likely, that these are formed by slow accretion. It's not all in one go, boof. So if clots form at different times and then accrete to make these macro clots, then clearly they can be different in different parts of the clot, and clearly they are different in parts of the clot in the sense of the percentage of amyloid. So when we get really down into the structures and start doing the proteomics, and then these techniques are available, so you can do this kind of thing, we will learn a lot more, but we're very early doors yet.
And where do you think these clots are being formed, especially if it's the accretion, where it's a slow buildup? It doesn't seem as though this is plaque disease, is it? I guess in theory, it could form in relation to a plaque, but it wouldn't be assumed naturally that this is forming somewhere, say in the heart. Say we know with atrial fibrillation, because there is stasis of blood, it's more likely to make clots, but you wouldn't expect it to make
that
this
is the
kind
party
of...
line. I'm going to butt in there, Philip, because
Yes, go
that is
ahead.
the party line, that the atrial fibrillation causes the microclots. Well, we've written a review which includes Greg Lipp, who's one of the big names in atrial fibrillation, to the effect that actually it's the other way round, and there's a lot of evidence that supports that view. So that the microclots cause the fibrillation, not the other way round.
Wow, no, that is a fascinating perspective. So therefore, the reason I ask though is that when we think about it, especially in the context of stroke disease, which is where you are looking at the samples that were collected, the two places, not the two places, but the main locations that these clots tend to come from are, say, for instance, the carotids, where there is plaque disease, and we would assume a break off of the plaque, which then blocks a blood vessel, but that should not look like amyloid.
That's the party line. The assumption was that it wasn't, and there's an assumption that a big lump of stuff breaks off and then goes up and causes the stroke, whereas our assumption is that actually it's probably a much slower process of accretion. Which is consistent with a lot of other things, such as the fact that transient ischemic attacks are precursors for strokes and a bunch of other stuff. But the to-do list is very obvious once you realize that this is probably the answer, to gather more evidence consistent with it, to do proteomics, to look at the relationship between. So Risha published years ago that post-stroke she can see the anomalous microclots, but of course it's not easy to prove that it wasn't that was just after the stroke you kind of shattered the clots you had into microclots, although the assumption is that isn't the case, that the microclots increase the tendency to have the stroke. But there are ways to find that out, both by longitudinal measures and other more specific measures.
I was curious, as you were talking, I was reflecting on something, and so this is just an exploration of thought. If we are producing, or if there are multiple microclots, where if you take a sample of blood, you are able to see microclots in the blood, you would think that all these microclots would then get trapped in the capillaries, do the damage, but that's the end of it. Why is it that these microclots continue to be formed ongoing? That doesn't seem to make any, I mean, it's so unusual.
So for any phenomenon, there's a rate at which something is produced and a rate at which it's got rid of, and when they get out of whack, the amount of stuff goes up or down. Now, in this case, we've got two competing processes, the normal clotting and the anomalous clotting, and we know that certain molecules will catalyze the anomalous clotting, and at tiny concentrations. So the first amyloid paper was one molecule of bacterial cell wall material called LPS for lipopolysaccharide per 100 million fibrinogens. Once you flipped into the anomalous form, all of the others go the same way, because otherwise they won't pack and make the clot. And it's similar with the prions. And before that, Risha and her team discovered that beta estradiol would also catalyze the anomalous formation. Now, the question is how big the clots are, how numerous, and all that kind of stuff. At those days, they were just looking qualitatively. But now we want to move into quantitative measurements to discover how quickly people get rid of them. Now, if you've got things that will catalyze it, free iron is another which we already showed, which is common in many diseases, especially if if a cell dies, its ferritin comes out, and its ferritin releases free iron. And we've written many papers on that. So there are many things that we know will catalyze the anomalous form, and you want to stop them coming into being too. And of course, some people are better at getting rid of clots, such as they have different genes for the amount of plasminogen activator inhibitor is one thing we know is involved.
You raised something there that again caught my attention, which is free iron causing or exacerbating clots.
Yep.
Based on what you just said there, one of the techniques that we use in terms of restoring iron levels in patients is to give them intravenous iron.
I know. I wouldn't.
No, really? So this is an important point, is that, so would you say that therefore in this context, if someone has that predisposition to make excessive clotting, giving them iron in the form of intravenous iron, which we use all the time, is likely to exacerbate that process?
Yeah, I don't think there's any doubt about it, because we've shown it in, in multiple times in many papers. So yeah, I know it's to do with the speciation of iron that is the issue. So people shove iron in because they think that people are short of it, but they're normally not short of it. There's lots of iron around, but it's normally held in a bound form, because the free form is actually very bad for you, because it'll stimulate the growth of bacteria apart from anything else.
Wow. Now that's important. The other question that I wanted to ask you, because you mentioned the bacterial endotoxins from the gram-negative bacteria triggering abnormal clotting. Follow this.
And we showed the equivalent in gram-positives, which is called lipoteichoic acid.
So follow this here. One of a paper that was done from Carlo Brogna, Italy, looked at the fact that abnormal toxins were being produced in the GI tract after COVID infection. Do you think this could be a contributing factor to the amount of inconsistency of microclots?
Well, we certainly know that the microbiome is heavily involved in all of these things. It's hard to study the microbiome because there are so many different microbes in it. And so if you have 1,000 variables to play with to produce an explanation, you can always produce an explanation, and 99.9% of the time it wouldn't be correct, because you have many more variables, i.e. different kinds of microbe, than samples, i.e. the number of patients you look at. So you can always produce an explanation when that is the case. And then it's, well, some years ago, we wrote a review about that too, called Statistical Strategies for Avoiding False Discoveries in Metabolomics and Related Experiments. So, but all of that said, in general, it is known that the microbiome is an important contributor both to health generally and to the likelihood of having bad forms of these diseases.
Wow. And so again, extrapolating what we've been talking about with these amyloid patterns, the mention that we had in prions, I made an extrapolation that I want you to challenge for me and tell me happily if it's wrong. I don't mind being wrong on this, but it was primarily to do with the issue of cerebral amyloid angiopathy, where we have deposition of amyloid in and around blood vessels, which weakens them, and then you have a collection of it, which then increases the risk of bleeding. My point was that if we are having any kind of prion-like increased amyloid pattern, this is the disease presentation that we are likely to see most commonly. Any thoughts about that?
It would logically follow. I completely agree. There is a particular version of well, essentially it's a stroke, isn't it? But a particular kind in which it is known that amyloids are formed, and it has this special name with amyloid in the name. So it's entirely plausible that that would indeed light up much more. I don't know the prevalence. It's not historically that high. I mean, high enough to have a name and be a well-known entity, but relative to the number of ischemic strokes, I think it's fairly low.
I think that, yeah, I think that what we're seeing now is the specific sequences that normally are able to identify these collections, these amyloid deposits, are relatively new in the past few years, where if you went back 5 years, it wouldn't be a typical standard part of an MRI investigation. So now that it is being done as a standard, we're seeing lots more of it. But yes, so it's difficult,
which of course
to,
doesn't tell you that the prevalence went up. But
exactly.
yeah, it's certainly a reasonable hypothesis that if folk like us are saying that, hey, amyloid is formed in all kinds of strokes, the fact there is one in which amyloid is known to form is not therefore a shock.
Yeah, these are quite significant patterns. This is why when I saw this paper, and when is it likely to get fully published?
Well, it's gone off for proofs. I mean, for printing. I mean, it's been accepted in its final form, and so I don't know how long that journal takes between the time of it going to there. But we were asked where we were in the next 7 to 10 working days or something. So hopefully it'll be out quite soon.
Yeah, and so in terms of this step, because now that you've got a bank of clots, and hopefully this bank will continue to expand, and hopefully there are other banks across the world that are doing similar kind of work.
There are, and we're talking to these people, of course, to encourage them to look themselves. We don't need to do it.
And what would you think about banks in terms of what is being pulled out from other kind of embolectomies, whether or not it is from pulmonary embolus, DVTs, if these were taken out? This fits in more with what the whistleblower in the US who had shown me, I probably won't show you all the pictures, they're a bit gruesome, but the pictures of these clots taken out of living people, why would these not be included in a bank? Why just
absolutely. Well, of course, the people who study the medicine is very subdivided. So the pulmonary people study the lungs, and the neurosurgeons study the brain. So they don't tend to get on each other's patch. So it requires people to talk to each other. But so we did this analysis of published proteomes of the amyloid clots and the normal clots. And then we did another analysis, was essentially asking the flip question. Although most people don't measure amyloids, lots of people have actually measured the proteome of clots removed from pulmonary embolisms and DVTs and the rest of it. And if you look at the proteome of those things, you can ask the question, are they more likely to be amyloid or normal? Because we know what kind of proteins are increased in the abnormal ones. And without exception, including the stroke clots, which we predicted, they were all amyloid, every one.
And this goes back for how far?
Well, we just analyzed published papers and did some informatics analysis on the proteomes. It's all published. You find it on my website. I can.
Yeah, no, that is fascinating.
Well, it's published this year. So if you look on my website, 2025, there's 2 papers. One where we analyze the proteomes, and the other where we make the predictions, including stroke.
Wow. This is fascinating stuff, and I really appreciate it, Doug. Is because not rarely, but it is so hard to get true scientists who are genuinely looking for answers, who are not afraid to look at anything that is in front of them, because that seems to be lost at the moment across the world. Because as I said, I've been asking about these embalmers clots for, people had to go into hiding to do the kind of research that was being done. They were so concerned about doing the exploratory science on it. We shouldn't, this is not right.
Well, science funding is hard at the moment. We managed to bootleg that off some money I had for a related project, but it certainly didn't say we were going to do strokes in it. But when you're set up to do the experiments, then it's quick and easy to do the experiments. As I say, all we had to do really was to having discovered the clot blank and talk to Alakenda, get hold of the things, give them to our histology people who sliced them up and prepared them. Then we do the staining in our automated microscope. So it was very easily done once we got that far. But it's hard. I mean, the thing we've gone into most recently, as you might or might not have picked up, is traditional Chinese medicine, which is definitely not seen as mainstream in the West. And the reason we have is I came upon this core concept in TCM and also Japanese Kampo, which is the same, and Korean Ryoyu, which is called blood stasis. And if you look at the name, and Virchow uses this too in his Virchow's triad, blood stasis is exactly what we're talking about. It's things that stop the blood going round as well as it should. So I started looking into this, and lo and behold, it's all of the diseases where we found microclots are considered to include blood stasis. And the more interesting part is that there is a treatment, a standard treatment for blood stasis, which is a collection of 11 herbs with the wonderful Chinese name Shui Fu Zhu Yu, Shui meaning blood in Chinese. And this is a cocktail that if you can't get your own practitioner, which is the best thing to do, helps cure people with diseases of blood stasis. And guess what? Although it's not an RCT by any stretch of the imagination, I know a lot of people who've looked at our stuff and are trying Shui Fu Zhu Yu, who've got long COVID, and guess what? They're getting better.
Wow. So this is going to be along the lines of something that happened with nattokinase or something like that, where it does help.
Yes, it's similar. And the nice thing about the herbs is that they scale. They're readily available. They're relatively cheap. And if 1 million people choose to take them up, they're not going to put a dent in the supply of Chinese medical herbs where there's however many 100 million people.
Wonderful.
So that's why we like this strategy, obviously assuming it works.
Yes. Wonderful. Thank you very much, Doug. Thank you for bringing this knowledge and bringing this science to the forefront. And we really look forward to that paper that you have coming out. As you go forward in your research, as a closing thing, can you tell us what is your vision for where this research needs to go?
So obviously we need to get some funding, which we're starting to apply for, and then we will look at the properties, obviously do a lot more people, do a lot more correlative staining. So there are standard stains that are done, but in the preprint we didn't have time to talk about that. There's a stain called Martius, and there's a standard stain called H&E, hematoxin and eosin. Look at all of those, do the proteomics, get more detailed analyses of, and Philip and I talked about this before the start, the various different kinds of strokes, and indeed TIAs, transient ischemic attacks. Just look to see the relationship between microclots, their composition, obviously the macroclots in the stroke, and the other embolic and related thromboses, to see how general this phenomenon is, what causes things to differ, and of course, more importantly, what's going to help people not have these things. If taking the Chinese herb stops you getting a DVT, seems to me like a good idea.
Brilliant, wonderful. So thank you very much for that wonderful education. And as I said, wishing you all the best in terms of your research, not just now, but in the future. Thank you, everyone.
Thank you very much indeed. As ever, much enjoyed chatting about science. We do like to do that as scientists.
Wonderful. Have a great evening. So just wait with me for the outro, Doug.